also the DSI website.
Thank you, Elizabeth. So let's begin with our webinar. And just a few thoughts. We as scientists and researchers bear in mind that the use of DSI underpins the work in all science fields. And that's why the parties at the upcoming COP15 must find a way to ensure the benefits of its use and that the benefits are shared fairly and equitable. There is an urgent need for further research into how to address and reverse biodiversity loss, which will depend on open access on DSI. So without further ado, I'll hand the floor over to Andrew Houghton, who will talk about what do we mean by open access.
Thank you, Marta. Just give me a second to put up my slides. All right. So thank you everyone for joining this webinar. My name is Andrew Houghton. I'm a scientific editor at Wiley. I lead the editorial teams for two journals, Advanced Genetics and the Biotechnology Journal. I have a background as a computational biologist and a real interest in open science and open data. And I have worked briefly on the topic of digital sequence information with the WildSeq project. So today I'm going to be giving a brief introduction to what do we mean by open access with DSI, so that before we get into policy options, hopefully we can have a common vocabulary to talk a little bit about what open means in this field. So before I get into the particulars of genetics and genetic data, I think it's always important to start with a very common principle of scientific communication. In science, you have to share your evidence. And this is a principle of scientific communication that is hundreds of years old. When you publish, you need to share your evidence and your data. Each scientific community has its own traditions and its own practices, its own way of sharing evidence and sharing data. And the genetics community and the sequencing community has its own particular history, which I won't have time today to get into in any detail, but I'm going to give you a very superficial, brief overview. And this particular history of sharing within the genetics community is created partially by the biology. So DNA is designed at a molecular level to be copied and shared. DNA is a molecule that your cells use to store information that tells the cell how to regulate itself, how to make proteins. And because it is basically an information storage media, it is very easy for this information to be shared and transmitted in computers and online. Now, because all life shares a common genetic heritage, the way that we learn from DNA is mainly through comparison. So whether you're a researcher who is studying all of evolution in a very broad sense, or whether you're looking at a single protein in a single frog, when you're looking at DNA sequences or other types of DSI, you understand them by comparing them to other sequences. And for this reason, researchers in the field need access to everyone else's sequences. For this reason, the genetics community and the sequencing community were very quick to recognize the value of sharing their data through common open databases. And already in the 1980s, three databases were launched: GenBank, the DDBJ in Japan, and the EMBL EBI in Europe, which host sequence data. They mirror their content, so this important genetic heritage is not owned by any one nation or one region. Data are deposited into these repositories at multiple steps alongside scientific publications with patent applications. So if you use sequence in a patent application, you have to deposit it to these public repositories. And some sequences are automatically deposited by major sequencing centers, whether or not it's used in a publication. The end result is huge amounts of sequence going into these repositories on essentially a daily basis. And these sequences are available to everyone. So we say that they're open, but what does that really mean? So for DSI, open means, first of all, access is anonymous, access is free of charge, and access is free of restrictions. And when I say free of restrictions, I think it's important to actually note here that the databases do not apply any additional restrictions to the data. This does not necessarily mean that there are not other restrictions, and I will come back to that point briefly in a minute. DSI are shared in ways that are interoperable and that support scientific transparency and reproducibility. Now, interoperable, it's kind of a complex word, but what this really means, a very common thing that for a biology researcher, they may need to download thousands or perhaps millions of sequences to perform even a simple analysis. So this means that the sequences need to be in common formats. There need to be common standards about how you talk about quality, and there needs to be legal interoperability to a degree, and that you have to be able to combine them together in order to do basic modern biological research. These sequences also form a central part of how we evaluate science. So it's a key part of how we as science talk about transparency and reproducibility. So this means that peer reviewers will often want to look at these data. When I am evaluating someone else's publication, I need to see their sequences. This is one of the reasons why access at the moment is often anonymous. With these huge amounts of sequences going into public repositories, digital sequence information use is complex, it's collaborative, and it's global. This entire sequence data set is downloaded over 30 million times a year, according to these main repositories. They can report that they have over 10 million unique users. And these databases are linked to thousands of other databases that share information. And these DSI are used by researchers in all countries. This graph here at the bottom is just emphasizes this complexity. This is showing data exchange links between just the EMBL EBI repositories and other external data resources which they are aware of. So this is just a small picture of this complex way in which data are shared. And many of these other data resources are serving specific communities, whether it's for a specific disease or plant genetics, etc. So before I take up more of your time, I just want to wrap up with two final points. So the first thing to really keep in mind when we're talking about openness and interoperability. Interoperability is so important because digital sequence information are often merged and combined and transformed along the chain of discovery. And so while scientists have very clear methods for talking about what they call attribution, whose data have you used, data transforms itself in complex ways, which can make, oftentimes you're not thinking about sequences individually, you're thinking about bulks, you're talking about mathematical models. The other thing is that open DSI databases, as I mentioned, place no restrictions on the sequences or their use, but this does not mean that they are necessarily free of all restrictions. Right, and so this is my segue into the rest of the webinar. Obviously, we will be talking more about what this might mean for benefit sharing, but a concrete example here, which is relatively obvious, of course, many of these sequences are going to be linked to patents. You may be able to download the sequence, use it in your analysis, whatever, without restriction, but that does not necessarily mean that you have rights to the technology that's been patented. So of course, there can be, so as you're thinking about using that sequence for commercialization, of course, you may have certain duties and other restrictions may apply. This is a complex topic. Thank you very much for listening to me. I hope this has been a useful introduction. I'm looking forward to the rest of the webinar. Here's my contact information. Thanks to Rodrigo Serra and Amber Schultz, who really helped me with a lot of these ideas I've shared in the past. And if you're interested in more, we have published 2 reports in the last year or two looking at openness in DSI. Take a screenshot of this or send me an email later and I can send you the links. Thank you very much for listening to me. I'm looking forward to hearing your questions and the rest of the webinar.
Thank you very much, Andrew. Now, our next speakers are Dr. Manuela da Silva and Dr. David Nicholson, who will be sharing with us an overview of the matrix that will enable parties and other stakeholders to analyze the different DSI options at the next COP15 this December in Montreal. Dr. Manuela da Silva is the General Manager of the Fiocruz COVID-19 Biobank for Human Biological Material and Viruses of the Brazilian Ministry of Health. Currently, she is the Vice President of the World Federation for Culture Collections. She's also part of the Executive Committee of the Global Genome Biodiversity Network and a member of the Ethics, Legal, and Social Issues Committee of the Earth Biogenome Project. Dr. David Nicholson is the Policy Advisor at the Wellcome Sanger Institute in Cambridge, United Kingdom. He has an interest in a number of genomic-related policy areas, including access and benefit sharing of digital sequence information, and he contributes to the Sanger Institute Research Center Culture Initiative by leading a project on equity in international collaborations. And before handing the floor to Dr. Manuela Silva and Dr. Nicholson, I would like to remind you that the Q&A box will be enabled at the end of the sessions for you to interact with our speakers.
Thank you very much, Marta, for the introduction. So let's share. Good morning, afternoon, evening to everyone. We are here to give an overview of the CBD policy options matrix and how do the policy options for DSI measure up. So here we have the performance matrix that was developed by the Informal Advisory Group on DSI in the context of the CBD for the multi-criteria analysis of possible policy options for benefit sharing arising from the use of DSI. So here you can see the 6 policy options and the criteria, sub-criteria. And this matrix allows policy options to be accessed against 19 sub-criteria within 4 main criteria that are effective in achieving policy goals, efficient and feasible to implement, enables good governance, and coherent and adaptable. So here we will present the approach that the DSI Scientific Network took to analyze the policy options and the results of the network's analysis, focusing on high scoring policy option. And then we will finish with 3 take-home messages. Here is our the DSI Scientific Network's assessment, the result of our group, the work we've done. And due to the ambiguity in the policy options at this time, we used a simple scoring system. So red for low performing sub-criteria that had significant problems or were not relevant, yellow for middle performing sub-criterion, green for high performing sub-criterion. You can see that we included a hybrid option here that is a combination of bilateral and multilateral benefit sharing depending on single or multiple DSI that were used and country of origin is known or not known. And we also want to present here 3 considerations from a research perspective. First, researchers upload, download, and use DSI largely from open, globally interconnected public databases. So, for example, there are no monetary costs or registration requirements to access the nucleotide sequence DSI from INSDC. Second, research and thus benefit sharing relies on the comparison and analysis of large volumes of DSI within the global data set. And third, research and scientific capacity building is often conducted through international collaborations. So such collaboration is greatly facilitated by the sharing of data globally. And these 3 considerations may explain the reasoning behind some of our analysis, particularly regarding criteria A that refers to effective in achieving policy goals. So this is the results of our work, and we ended up with 3 high scoring options. The option 3.2 that refers to other payments and contributions. So benefits are paid into a multilateral fund for benefit sharing that is distinct from the option 3.1 that refers to payment for access to DSI. And in this case, we have focused on a sub-option involving cloud-based DSI models. Another high-scoring option is the option 6 that refers to 1% levy on retail sales of genetic resources. So we will have a multilateral fund financed through a levy on retail sales of goods arising from the use of DSI and or physical genetic resources. And in this case, we have assumed that the multilateral system would be the rule and the remaining bilateral procedures would be exceptions. And another high-scoring option is the option 4 that refers to enhanced technical and scientific capacity and cooperation. For example, research collaboration, training, knowledge platforms, technology transfer, technological development, database infrastructure. And in this case, we have assumed that there would not be enforcement requirements for providers or users. So now I give the floor to David, who is going to continue the presentation, and thank you very much for your attention.
Thank you, Manuela. So I will now take us through the DSI Scientific Network's assessment of these 3 policy options. So I will start with criteria set A, which looks at whether the options are effective in achieving policy goals. So firstly, in all 3 of these options, benefit sharing would be decoupled from access to DSI, and therefore access to public databases remains open and research and innovation is not hindered. And indeed, research and innovation may be supported, especially by option 4, which includes capacity building. And because of this, benefits will continue to be generated, and therefore these options have potential for benefit sharing, with the caveats that option 3.2 does not focus on non-monetary benefit sharing, and option 4 does not address monetary benefit sharing. And finally, all of these options have potential to contribute to conservation and sustainable use of biodiversity, and option 6 explicitly refers to this in the description of the option. Next slide, please. So I will now move on to criteria set B, which looks at whether the options are efficient and feasible to implement. So we scored these options lower in this criteria compared with criteria A, C, and D, but they still scored higher than most of the other policy options here. So one potential source of difficulty is that for options 3.2 and 6, external entities would need to be involved, which may affect feasibility of implementation and increase administrative complexity. It is also unclear how quickly parties could implement these options in practice, especially if national legislation is required. And finally, option 3.2 and 4 do not directly address DSI use, and so they cannot easily be used to distinguish commercial from non-commercial DSI use. And next slide, yeah, thank you. The third set of criteria look at whether the policy options enable good governance. So firstly, options 4 and 6 are relatively conceptually simple and easy to understand, whereas some might consider option 3.2 less straightforward. Secondly, providers do not have direct control of enforcement in a multilateral system, so all 3 options score quite poorly against subcriterion 14. However, these options do score well for ease of compliance and avoiding jurisdiction shopping. And this is because decoupled multilateral systems put no restriction on DSI access from any jurisdiction, and they also place comparatively less burden on researchers compared with some of the other options. And finally, benefits that have been collected in a multilateral fund could be purposely directed to Indigenous peoples and local communities. And next slide, please. So the final criteria set looks at whether the policy options are coherent and adaptable. We've scored all 3 options highly here, as we believe multilateral approaches have the potential to be part of a wider coherent framework, and decoupled multilateral approaches could capture a broad scope of DSI and account for technological developments such as machine learning algorithms, which may be trained on DSI but do not themselves contain DSI. So next slide, please. So before I finish with some conclusions, I just want to acknowledge that we only had time to explore 3 of the 10 proposed policy options in this presentation. So I encourage you to visit the DSI Scientific Network's website to see a more complete analysis. So our first message is that options that decouple access to DSI from benefit sharing would not compromise open access and would support research into conservation and sustainable use of biodiversity and generate benefits, whilst offering the potential for fair benefit sharing. And how successful and equitable that benefit sharing would be likely depends on how these options are implemented in practice. Our message 2 is that decoupled multilateral benefit sharing options recognize the realities of the scientific DSI ecosystem, where sequences are stored in global data sets and are used, compared, and analyzed by researchers around the world. However, even for these multilateral options, there are still some challenges, some significant challenges regarding implementation, but we suggest that these challenges are fewer than for policy options that do not recognize the realities of the DSI ecosystem. And the final message, and the final message is that a combination of multilateral policy options might be the best approach, so that the relative strength and weaknesses of different options can complement each other. And analysis of the policy options using the matrix, as we have done, may be a good way to identify how this might be done. And next slide, please. So that's where we'll finish off. So I'd just like to thank you very much for your attention. Please don't hesitate to contact the DSI Scientific Network via the website, or Manuela or me through our emails if you have any comments or questions that aren't addressed in today's session. Thank you.
Thank you very much, Manuela and David. I would now like to introduce Derek Eaton. Derek is Senior Director of Public Policy Research and Outreach at the Smart Prosperity Institute at the University of Ottawa in Canada. Derek has a wide global experience across many sectors, including energy, agriculture, finance, and innovation, where he has played a leading role in the development of the international agenda on biodiversity, climate change, and the green economy. Today, Derek will walk us through the economic implications of the DSI policy options. Welcome to the floor, Derek.
Thank you very much, and hello, everybody. I'll just get to the screen, share my slides. There we go. Yes, just perhaps as an additional explanation, the Smart Prosperity Institute is a think tank and research network. We focus on aligning economic policy with both climate and nature challenges, sustainability challenges. I am based myself in Toronto, and I'd just like to acknowledge that I work and reside on the lands, the unceded traditional and ancestral lands of many First Nations peoples. That includes the Mississaugas of the Credits, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat. And at Smart Prosperity Institute, we recognize the unique and enduring relationship that exists between indigenous peoples and their traditional territories worldwide. So our work that I'm going to go through very shortly today is part of the WILDSI project, where we have been thinking through some of the economic implications of the policy options for the exchange of DSI. It's been a team effort, and you can see my co-authors on this slide, and my email address is there as well. And you can find our website quite easily if you Google Smart Prosperity Institute and get in contact with me if you didn't get a chance to note that here. So when we initiated this project almost a year ago, what we really wanted to do was to conduct an analysis of the costs and benefits of the alternative options. And this was to, in a sense, replicate work that I had been involved in, perhaps I think about 20 years ago, in doing a similar type of exercise in advance of the negotiation of the International Treaty on Plant Genetic Resources for Food and Agriculture. We found, however, that there is so little clarity and detail on the modalities of these options for the most part, that this was really not feasible in a very concrete way. So we wondered, well, why is that? Why is it so hard to imagine what these options really look like? And yes, there are a number of specific design issues, but we realized that there's perhaps a more fundamental issue to thinking about the economic perspective on DSI. So we've moved more towards developing a framework to identify and assess the various risks and opportunities. Those can be thought of also as costs and benefits, respectively, that exist in DSI marketplaces. And then, if we can, to apply that to policy options. The first thing that I would really like to emphasize is that in thinking about the framework, an economic framework for DSI or the DSI economy, we have to recognize that DSI is not a traditional physical good or material. We understand how easily, costlessly it's replicated and shared, and it really just does exist at a certain point as a digital stream of bits and bytes, zeros and ones. And this led us to think much more about the economics of DSI in terms of many lessons and ongoing insights developed around the economics of information more generally, which has particularly exploded with the rise of the digital economy, or as we would say, DSI being part of the digitalized economy. So that's the first key message we have today, that DSI should be thought of as part of the digital economy. This is a representation of thinking about different sort of areas of the digital economy that has been discussed in fora such as UNCTAD and the OECD, where there's a distinction made between the core digital IT and ICT sector. We all think of that typically when we think about digital economy, and a middle area in this red circle, which captures the rise of digital services, and in particular, the platform economy. But if we look at what's happening across actually many sectors of the economy, there's a digitalization process underway in varying degrees, and in varying degrees according to sector, but also depending on where we are in the world. So these technologies are having a very disruptive effect on a number of sectors, including, for example, agriculture, health, and pharmaceutical. And so we would propose to think more about DSI as falling within this broader scope of the digital economy, which, as I say, emphasizes, goes beyond our thinking about the digital economy in terms of the very popular and almost ubiquitous consumer-facing platforms for video content such as Netflix and what have you. And so a second key message that we're taking away from our work is to think about the importance of platforms. The platforms are key in all aspects of, pretty much of all aspects of the digitalized economy to facilitate and flow the use of information. And there are transaction platforms. Those are more typically the type that we see, again, with the consumer-facing platforms and subscription-based services such as Netflix. But the digitalized economy, and particularly DSI and where it's used, where it's applied in other sectors of the economy, is based more on innovation platforms. These provide ways for sharing common designs, archetypes, and interactions across a sector. The third key message or principle almost that we see emerging in this work as we look at DSI as part of the digitalized or digitalizing economy is that policies that promote frictionless exchange generally have the potential to provide the greatest economic value, or perhaps in terms more familiar to the CBD, to generate the most monetary benefits and non-monetary benefits to the extent that those are economic as well. So these points to principles that policies should bear in mind, such as the importance of free, open, and interoperable exchanges, the fair and equitable flow of information. Also, not to be forgotten, the importance of privacy protections where relevant for individuals and sector actors, and the overall purpose of supporting economic growth and innovation. Finally, as a key message, we see the importance of decoupling exchange from revenue generation. This occurs actually even in subscription-based services in the more core digital economy, to which I was referring to. But in order to generate benefits, and effectively the largest benefits possible, this decoupling of the revenue generation is it's a pattern we see in many places, and the fundamentals of DSI, just given the scale, really reinforce that. As Andrew's mentioned already, there are multiple platforms distributing DSI, which further provides reason for the importance of this decoupling to support the comparison and recombining of DSI. And also, this decoupling is a critical part of reducing transaction costs. So as we, just to my concluding slide, as we think about what our framework implies for the policy options under consideration, it seems fairly clear that the more bilateral types of options, including sort of hybrids approaches that are being discussed, really have a strong risk of decreasing this free and frictionless exchange and are likely to considerably reduce the generation of benefits from DSI, both monetary and non-monetary. Options that suggest a payment for access, such as 3.1, would most likely considerably decrease cross-platform use and the research with DSI, and thus the generation of benefits. And then options that are based more on other decoupled payments, such as 3.2 or potentially 6, provide the best opportunity to maximize the benefits from DSI. And of course, this, in order to ensure a fair sharing of the benefits, this underlines the importance of the design of a revenue generating mechanism that will work towards that end. So I will conclude there, and thank you very much for your attention, and look forward to questions and exchange. It's a pleasure to notice in the chat a number of old acquaintances or colleagues are present here today amongst everybody. Thank you very much.
Thank you very much, Derek. And a note to all the participants, if you would like to ask Derek questions or any of our panelists, please note that the Q&A box is now open, so direct your questions here. I'll now hand the floor over to Sarah Hearn and Christine Pratt for 2 case studies that will highlight the use of DSI in research and the role of open access.
Hello, everybody. Good morning, or good morning for me. Good day to those of you in other geographies. Just let me get my slides set up. So my name is Sarah Hearn. I'm a project scientist and PI who works at CIMIC, that's the International Maize and Wheat Improvement Centre. We're a member of the CGIAR, and as part of that, we are heavily involved in generating and using a lot of DSI. So the CG is a network of international research centres focusing on the improvement of agricultural systems in the developing world. And as part of that, we hold a number of international germplasm banks, 11 germplasm banks in total. And these gene banks form the largest and most frequently accessed network of gene banks globally when we look at materials that are distributed under the international treaty. So these germplasm banks together hold around 750,000 accessions of different crops, which form the basis of human diets globally. That's around 10% of the ex situ collection. And what I'm going to talk to you today about is a series of work that we've had that we now call under a large umbrella, the Allele Mining Initiative. So this work stems from 15 plus years of work in the space, which has really been focusing on the use of DSI as a tool to better identify and leverage value from these extensive germplasm collections, with a focus on using that genetic variation, that native variation, in targeted crop improvement. So the Allele Mining project focuses on the identification and leveraging of high-value genetic variation, with a particular focus here on climate change adaptation. And the current project that I'm going to talk to you about focuses on a multitude of commodities that are held in trust by the CGIAR. And this is predominantly funded by the Bill and Melinda Gates Foundation and FFAR, aligned with the AIM for Climate initiative. We're listed as one of those sprints. So AIM4C was an endeavour announced at the last COP26, which is a collaboration between the USA and UAE, really looking at how we mitigate the impacts of climate change. So when we think of these germplasm collections, when I walk into the gene banks, as a user of genetic variation, as a user of germplasm bank accessions, this is predominantly what I see. We have many, many flasks and collections that sit on the shelves of the germplasm bank, but much of the value in that variation is hidden. So I use the analogy of this being rather like walking into a supermarket and seeing lots of cans and products on the shelf, but not really having any other information to make an informed selection out of those tens and hundreds of thousands of products, which ones may be most relevant for use for a particular breeding challenge. So the breeder is rather like this poor gentleman here, sat looking for a needle in a haystack without any information or any magnet to try and find that resource more informatively. So what we're moving towards is using DSI as a framework to help provide some common standardization so that we can access alleles, haplotypes, and other forms of genetic variation that have breeder relevance in a much more data-driven manner. So the DSI helps provide a common framework across tens and hundreds of thousands of accessions, which enable powerful scientific investigation to be conducted and enables clients like breeders and other scientists to access potentially more relevant variation rather than this guesswork that's currently going on. So this is what some of the collections look like. Here are some beautiful examples of maize, which is what I'll focus on now. So here we can see color, we can see seed shape, but that doesn't tell me if any of these materials may be drought or heat tolerant. However, what the materials do have by and large is a passport data, and this is just like your or my passport. And that details in many cases where these materials have been collected. Now, this distribution, in the case of the germplasm bank collections, broadly gives very good reflection of the centers of origin of these domesticated crops and the centers of diversity of the crops. And as you can see here, looking across some of the collection from the CIMMYT gene bank, and these are just those materials that have well-detailed geolocation in their passport data, we're covering a very broad range of environments. From this geolocation data, we're able to pull out long-term climate variables from the collection sites. So this helps detail the environments under which these particular land races have evolved, they've been grown and selected by farmers for properties of value. Now, this indicates that we're able to sample different niches, so niches that reflect that natural and farmer-based selection. And what the allele mining work does is take advantage of this information together with high-density genotypes, DSI-based generation, to run a kind of association analysis. So we take the paradigm, if a material has been collected from a hot and dry environment, it should have allelic variation which gives that material an advantage in that environment. And together through standard analyses, we're able to identify regions of the genome and alleles and variants therein, which provide and contribute to different levels of adaptation. So here, for example, this is a scan showing chromosome 9. As we move along here, we're walking along the chromosome. And this little tower here shows me that there is a region here that is associated, in this case, with low precipitation in different environments, low rainfall, potentially some level of drought tolerance. And when we look under this particular peak here, we find a gene that has known function, but may be a favorable target for breeding to introduce this variation, which may not have got through that breeding bottleneck. So as breeders have taken land races over the years and have improved those into the modern cultivars we see today, a lot of the good variation never made it through this bottleneck. And so we're able to go back into the collection and start to identify some of this variation in a targeted way and use that effectively in breeding. So we're starting to look at this across a range of mostly abiotic stressors, such as drought, temperature stress, acid soil, salinity, and then other soil-related features and agronomic use. We're able to take DSI and equally use it in a slightly different paradigm. So many plant breeders will have heard of predictions and a paradigm called genomic prediction. And in that paradigm, normally what we try to do is predict the value of a known genotype. And we've got a potential new variety and we try and predict its value from its DSI data. Here we take that paradigm and kind of turn it around on its head. So we take the same climatic data that we have from these geolocated genotypes. We use the same DSI data and we develop what is commonly called a training panel. It helps provide information of how the climatic variables and the genotype are linked together. And then what we do is take a potential environment of interest. So this could be a particular high value site where we are growing a particular commodity at the moment. And we can look and say, okay, this particular market is going to have this environment in, say, 20 years' time. We can use the climate modeling to predict an environment. And then we use that environmental feature, that environmental characterization, to ask of our training panel, what would be an ideal genotype for that particular environment? Now, in this case, that genotype may not exist in reality. It is a synthetic construct that we are making and saying, if a particular entry looked like this in genotypic terms, it should have higher adaptation and potential value for breeding in that environment. And we tested this paradigm with some known data. So here, what you see on the x-axis is the distance of entries that we have actually phenotyped in the field from the ideal predicted genotype. So as you move from this point, you move to the right, we're moving further away from the ideal genotype. And as you see here, this is a measure of performance of the material. In this case, it's grain weight, it's yield under a drought-stressed environment. And you can see that the materials that have closer genetic relationship to the ideal genotype have better performance. So this is a resource that we can use in our future breeding and predictive work. Now, one thing that I will say here as a large caveat is all of this data analysis is based on use of tens and tens of thousands of data points generated from a few up to 17,500 accessions. So the value of this comes from analysis across a broad range of materials that have been sampled from a very broad range of environments. So we have, without the implementation of political boundaries, a very good representation of the evolutionary history of the particular species. So to finally wrap up, I just want to talk about how we've been managing data under this project. So this started off from 2011, from an earlier project that was working in this space. And we had a series of meetings with the various stakeholders that were involved within this effort. And the key drivers here were that the stakeholders wanted to ensure that data remains in the public domain as an international public good, and to ensure that data was as accessible as possible to a broad range of stakeholders. So on the second element, we have, and the CG continues to have quite extensive activities in the capacity development space, trying to ensure that stakeholders have the ability to access and use data in as appropriate a manner as they require. But on the IP related issues, we have to date, with all of the data that is generated, we release that data with data licensing. And that material and that information is shared via CIMMYT through our own instances of two platforms. One is the Harvard Dataverse, and the other is Germinate. So Dataverse we use more as a flat file sharing platform. Germinate is a relational database which provides a little bit more information. So the licensing here really has elements that focus on the use of data freely for research, educational, and scientific publication purposes. But to address many of the concerns that the broader stakeholder community had about the risks of particular elements of native variation being removed as international public goods for use, we have got a particular line in there that requires the users not to seek forms of intellectual property rights over the data that is accessed from that platform, and that the data may not be redistributed to other platforms without written consent of CIMMYT, because that can remove that license application. So the data and derived knowledge is available in processed and raw forms, depending what particular users and stakeholders want to do. The licensing, unfortunately, due to some IT and technical issues and challenges of machine harvesting, the platforms do require users to register and to log into access, but that registration is not policed, the access is not policed. We simply don't have the bandwidth or the ability, the resourcing to do that. Now, this licensing has been put in place as a stopgap solution. Yeah, rather than just ignore the many significant concerns that the stakeholder community initially had, we put this in place while we wait for definition and implementation of global DSI policies. So personally, as a hardcore generator and user of DSI, I would favor the flattest and least intrusive approach for DSI management, one which obviously reflects some of the concerns about IP over native variation, but one which has the lowest possible transaction costs for both generators, curators, providers, and users of DSI. And this is a view that is broadly shared by my colleagues in the CGIAR. Okay, so thank you very much for your attention. And I now hand over, if I can escape my presentation, to the next speaker. Thank you.
So hi everyone, I'm Christine Pratt. Today I'm speaking from the south of France, from Marseille, and I am the operational director of the European Virus Archive, which is the largest globally active biobank of viruses worldwide. And I'm going to share my screen just a few seconds. Okay, you should all be seeing that. Is that correct? And okay, so I'm going to talk today about the use of DSI for viruses and how access to viruses is quite fundamental. Let me just try something else, sorry. Just a second. Okay, that's much better. Okay, so I'm going to share how the sharing of viruses is quite fundamental for dealing with viruses. So the European Virus Archive, what it is, it's a non-for-profit organization. It's a European research infrastructure that groups 46 virology labs worldwide. So here is a little map of where all our labs are located. And our missions are just summarized in this little graphic animation, basically. We observe viruses, we isolate them, standardize them, and then we ship them to anyone that needs to be working on them worldwide. So to do that, we have an online catalog that is called the European Virus Archive, and scientific worldwide can request any virus they need to work on. So when there's an emerging virus that is causing an outbreak, there are many, many different questions that are asked, basically. What's the transmission of the virus? What's its genome like? What's its distribution like? What are the symptoms caused by the virus? What are the dynamics of the epidemics? What is the sequence, etc.? Lots of questions of a virus that is by definition completely new and unknown. So there's basically an urgency to gather information on this virus, right? And you probably have heard a lot this term in the recent few years because of COVID-19, but What does it mean when we talk about preparedness? How to be prepared for new viruses? What is the main objective? The main objective basically is to reduce the response time, when a new virus arrives. And what does it mean in terms of practice? So in terms of practice, we need to be able to diagnose the new virus. So we need, for example, positive control for the molecular detection of the virus. We need reference material to check that laboratories are prepared to diagnose the new virus. We also need what I call therapeutics, like, for example, antivirals, vaccines, strains. For that, we need strains. We need strains of viruses to check that the products we're developing are actually useful against this virus. And we also, in terms of research, we need to have as much information as we can. So we need genomic material, biotechnology tools, etc. And for all this, for diagnostic kits, we need them in terms of hours or days. We need them super rapidly. For therapeutics, I mean, you know, ideally we would have them like the next day, but of course we can wait a bit longer because they take longer to develop. And as for research, I mean, the time frame is a bit longer. So we're talking days, weeks, or even months. But for all this, DSI are used. And I'm going to present you basically the case study of what happened with COVID-19, right? So on this slide here, I have represented, it's a timeline, representing in blue the different epidemiological news, and in orange, the different actions by EVA. So on the 31st of December 2019, China reported an outbreak probably due by a novel coronavirus. On the 10th of January, they released the genomic sequence of the SARS-CoV-2. So the new virus was named SARS coronavirus 2 because it was identified as belonging to this family. And on 23rd of January, so very soon after, the first autochthonous cases happened in Germany, in France, and later on, Italy was placed in confinement on the 10th of March and France on the 17th of March, 2020. And at the European Virus Archive, one of our missions, as well as supporting research, so what I would call a bit of a routine activity to support research on viruses, one of our missions is also to be there and to respond in case of viral outbreaks. So 4 days only after the genome sequence of the virus was released, our colleagues from Charité in Berlin, who are notoriously known for being, you know, experts on coronaviruses, 4 days after, they developed diagnostics positive control that were recommended by WHO and placed on the EVA catalog. And they used DSI to do that. Then our colleagues from Erasmus Medical School in Rotterdam developed an RNA specificity panel as well to make sure that the labs were prepared to diagnose the new disease. They used DSI as well. And the first strains were added to the catalog, and soon after, so less than 1 month after the sequence was known, a proper diagnostic kit was developed by the biobank and made available. So you probably all remember this trilogy that we heard a lot at the beginning of the pandemic. It was called at some point the COVID Tetris game. I don't know if you have the reference or not, but basically the idea was to test, to trace, and to isolate. So those three were like the mantra that were used to try to control the pandemic. And I've represented here on this graph what happened for the European Virus Archive by representing the number of orders per week for the diagnostic kits that we developed in the biobank. And so basically, one order allows to run around 10,000 diagnostic tests. So you can see that on the week number 4, we had 250 orders already. And then 3 months, during the first 3 months of a pandemic, it was possible basically to run more than 14 million diagnostic tests in the next 3 months after the release of the genome sequence, thanks to the distribution by the biobank. So now we'll go into more details of how the diagnostic kit was conceived, and you will see that it's very routine for us, but general public might not know that. So I've highlighted the parts that are super important, and they are directly extracted from the publication by the Charité team in Berlin. So basically, they basically relied on social media reports announcing the detection of a SARS-like virus. So they assumed that a SARS-related coronavirus was involved in the outbreak. Then they downloaded all complete and partial sequence available in GenBank. Then they did a manual check, and as you may or may not know, those banks contain what is called artificial sequenced, for example, laboratory-derived or synthetic sequences that are not directly extracted from viruses. And they removed artificial sequences and sequence duplicates from that analysis. Then they did an alignment of sequence that was completed by any additional sequences released independently on GISAID. So that's what it looks like, basically. So here they have presented in this figure the alignment of different viruses from coronaviruses, for example. So the human Wuhan outbreak sequences, but also bat-related SARS coronavirus or the SARS-CoV-1 sequence, etc. And they aligned all the sequences, and that allowed them to choose which part of the genome they would need to target in order to develop a diagnostic kit for that. So in conclusion, the conclusion that I want to stress out that it was preparedness work. So they assumed that it was a SARS coronavirus that was involved in the outbreak, but it might have been something else than SARS. But for the sake of preparedness, they accessed the sequences and aligned them and designed some diagnostic kits just for the sake of preparedness. They also used artificial sequences that were not used at the end in the design of the specific diagnostic kit, but still they were accessed in the current ABS sense. So I've summed here up the different consequences of different policy options. If we're talking about decoupling access to DSI and benefit sharing, it guarantees open science, as my colleague Andrew presented earlier today. It allows for capacity building globally. It is much more time efficient, and it allows for trials and errors, as I've shown you. Trials and errors are a big part of how science works. And then for the hybrid bilateral and multilateral solutions or bilateral solutions, it would slow down the access to resources. We saw here that the diagnostics was made available in a matter of days, and that allowed countries sometimes to be able to diagnose the first cases of the people landing with the coronavirus disease on their land. It would limit the distribution of the material. There would be a risk of political and economical polarization of exchanges in times of crisis, and there would be less scientists working on developing countermeasures, probably. So how the current DSI model generates benefit sharing, and I'm going to present the EVA model during the COVID-19 crisis. So the European Virus Archive model. So we had basically 12 providing countries providing providing DSI material, and they distributed to 97 beneficiaries countries. 33% of them were distributed free of charge. The rest was distributed to companies that had to pay for the access. And 53% of those accesses were to low and middle income countries. If you want to know more, I invite you to read this publication that was published in 2022. But we had a lot of non-monetary benefit sharing. So we shared sequence data, we shared material, we initiated collaboration in research and development project, and we also shared or transfer of research and development results such as trainings and equipments. And that's the end of my presentation. So I will stop the sharing of the screen. And I think I'm giving the floor again to Martha.
Yes, thank you very much, Christine and Sarah. So I would like to thank our audience. We have many people in our audience from different parts of the world interested in the topic. And the Q&A session of this webinar is now open. I would like to remind you that the Q&A box is enabled. You can ask your question to one panelist in particular or to all of them. It's been really busy, so we have our team from the DSI network pointing out some of the questions. For sure, we are going to have questions and comments as an input for our work on dealing with this topic. So we will start with a question for David. David, will the multilateral system be cheaper to run than each country adding to their own existing the Nagoya system?
I suppose that's a difficult question to answer without knowing precisely which multilateral system it's going for. I think there's also an important consideration which the other presenters talked about around the fact that the multilateral system will generate more monetary benefits and non-monetary benefits, and therefore, along that line of logic, that more money will be generated. And yes, I think one of the main disadvantages of having a bilateral country-by-country system would be the fact that those monetary and other benefits will be, less of those will be generated.
Thank you, David.
Thank you. I can ask the next question, and that's to Manuela. So the question is, it's written in Spanish, so I've had a help with the translation here. So what would be the main feature of the hybrid option that would generate the least benefits, monetary and non-monetary?
Thank you, Elizabeth. Thank you for the question. The fact that there is no clarity when it should be through the multilateral mechanism or through the bilateral mechanism transform it in a very complex situation. So this option may bring more bureaucracy and complexity, which may make difficult to get the benefit sharing from this. I hope to have answered.
Thank you, Manuela. Now up to Derek. Derek, could you define the term decoupling? How does it relate to lowering transactions costs, and how does it relate to management of downstream IPR value?
Yes, certainly. Thanks for that question. And I think there was another, it relates to a question that I think also was asked maybe by Joseph Fogel around just the word decoupling. And so decoupling is for the team that I've been working with on this economic framework. This one we chose. I'm not necessarily with a very I don't know, should say thorough thought process about what was best. We think we thought at first instance it's helpful in terms of communication. So the question is being posed maybe suggests that it's not so helpful, but let me try to elaborate a little bit. I mean, we're talking about the decoupling of the access to DSI, the exchange of DSI. So transactions involved, we call it transactions or exchange of DSI, decoupling that from monetary revenue generating mechanisms or payments. So that's something that's quite common in many areas of the digitalized economy. And it relates to, in part, to the transaction, well, largely, you could say largely to the issue of transaction costs. When you have a high volume of sort of exchange, let's call them exchanges. So, somebody exchanging some quantity of DSI with a scientist with another scientist, or accessing a database. I mean, when you have a high volume, if every time that action is undertaken, it requires, some procedure to be followed that, in order to register for a monetary exchange or a fee, then that's a transaction cost. So the strong rationale for this decoupling is that there's a high volume of exchanges, and also with very different types of potential economic values attached to them. So it would be the decoupling helps ensure that those exchanges actually take place, where the transaction costs that would otherwise be involved would likely discourage many actors from even engaging in the first place. So I hope that's clarified it a bit, but I do appreciate the question and that it's a term that can lead to confusion. Thank you.
Thank you, Derek. The next question I would like to direct towards Andrew. So the question is, by which way, and especially how much a given local community or Indigenous people could claim from a decoupled multilateral DSI fund if they or anyone else in the ABS system don't even know what their contribution to the DSI system has been?
So this is a really good question, and it's not an easy one to answer, because I think it's hitting on the nose one of the challenges to any solution to this problem. And one of the points to keep in mind is that when we talk about DSI from an Indigenous community or a local community, what does that mean? Does it mean it's geographically from the region where they live? Does it mean that it's a species that they have a cultural connection with or a spiritual connection? I would argue that any of those definitions are valid and deserve to be respected. Obviously, countries are going to have this same concern, right? So if we have a decoupled system, how do I know what sequences are coming from my country? Countries are a little bit more geographically defined. So we have proposed that some of the people working in the DSI network and in an Asia communications paper that I and some other people have contributed to, that again, we're focusing on a decoupled solution because that's going to generate the most benefits. But then you're going to need a system to figure out how to distribute that money. And that's going to need to be a process that takes into account different types of information. You can get good geographical information from the public data sets, and there's proposals to improve that information. But again, we're not suggesting that you try to track every individual sequence, just that you could use that as a guide to help you distribute money, right? And there are also good proposals for putting Indigenous tags on sequences as part of scientific metadata. And again, I think that those are fabulous ideas. But we shouldn't confuse the sort of scientific metadata with a track or trace solution. So this is have a decoupled solution that generates the most benefits possible, that loops in machine learning, like David said earlier, things that aren't easy to track and trace, and then look at ways to guide people with good analysis, good information. And hopefully we can respect some of these different ideas of ownership and allow countries in that solution to have some flexibility rather than a formula right up at the front. I hope that's useful, but I think that this is a really hard question to answer right now.
Thank you, Andrew. Okay, now for the next question. This is for Sarah. There is an illustrious literature in economics that distinguishes use value from exchange value. It becomes especially important when considering products that are essentially information, as unbound competition makes production of such information uneconomic. Hence, limited in time monopoly, intellectual property rights are one solution. When you speak of value for for what it is essentially natural information, are you referring to the use value or the exchange value?
Okay, so I can reflect the feelings of the stakeholders that we had consultation with. So use value is was the most immediate and probably the best articulated concern by stakeholders. And these stakeholders, by and large, are not economists. Yeah, I'm not an economist. They're not using that kind of terminology. But use value is the most immediate concern. However, exchange value was also articulated. So we are covering both elements there. It isn't one or the other. So both do need to be considered. And there is almost an allergic reaction amongst many of these stakeholders when we talk about putting any natural variation under any particular intellectual property agreements. Yeah, it's a very hot topic, and I think one that needs to be addressed suitably with appropriate sensitivity.
Okay, so thank you, Sarah. I have the next question, and I would like to ask a member of the panel to volunteer to answer. So I'll make it to the whole panel to start. The question is, I wonder if the reflections include the fact that payment for access could restrict access to DSI databases to the most funded scientists only and to private companies, which would increase inequality between scientists, even in wealthy countries, because the level of funding can be very different, even in the wealthy countries. So maybe I could, Christine, if you could start maybe? Or if another member of the panel would like to volunteer?
I can speak briefly to that topic, although I just spoke. But so I think that most of the members of the DSI network and others are not in favor of a subscription fee on access for exactly this reason. So most of the suggestions that I'm in favor of are some other sort of decoupled payment that might be connected to commercialization, product sales, or national contributions. So I think we're all looking at ways to make sure that people can play with the data and still maintain that open culture. So that is, but I would like to just point out another issue here. In my view, there are certain people who are studying virus evolution, people who are looking at biodiversity monitoring, who need much bigger data sets compared to some types of biotechnological applications, like where you may need a very focused data set for a specific enzyme. So we also don't want to be disadvantaging big data heavy fields that aren't commercially relevant. So I think that's really important as well. And that's, I think, why a lot of people are not in favor of a subscription fee solution.
Would
anyone else? Yeah.
Okay, so let's go with the next question, and it's also open for our speakers. Is there a way of knowing who has access what, especially when it comes to DSI, that can be used to make biological weapons?
So no, as I said, at the moment, no, there is no way of tracing that.
Okay. Let's keep going with another question.
So I have another question for the panel, if someone would like to volunteer. What DSI contributes in biodiversities in some countries? So maybe I can rephrase that. So how will DSI contribute to biodiversity in different countries? Manuela, you've raised your hands.
Yeah, thank you. Well, let's start from the beginning. It helps to know our biodiversity. Thanks to molecular taxonomy, we can identify our the representatives of our biodiversity. And with the development on biotechnology, then all this is reverted to the conservation, just for a start. Thank you.
Would anyone else like to comment, follow up? Sarah?
Yeah, I mean, rather than broad scale biodiversity, I can talk to specific biodiversity. So we have used a lot of this information in relation to the collections that we hold to try to identify where there are particular gaps in collections, to prioritize collection of material from areas that may be under risk, whether that's climatic risk, whether it's political risk, or risk in terms of people abandoning agriculture. And again, we use that information and have used that information to see what diversity was available or being used in farmers' fields in the last century and what is being used nowadays. And we can use that to many regards to try to anticipate potential risks from diseases and other things like that.
Thank you, Sarah. Now I have another question for Christina. Christine, how sharing of avian influenza virus with the WHO was delayed according to the provisions of the CBD and Nagoya Protocol? The sharing of COVID germ also could have delayed for any potential monetary benefits. What would have been the impacts to humanity if there happened any such delay in sharing of the COVID germplasm?
Well, so basically, we could imagine, I don't have numbers in mind, obviously, but we could imagine the pandemic to go undetected for much longer in that case, and because simply people would not be able to diagnose it molecularly speaking. And as we know, COVID-19 has many, many different forms. It can be like hospitalization, but it could also be asymptomatic or mild symptoms. So detecting it just only based on symptoms is highly challenging. So you need a molecular detection at some point with diagnostics. So delays in sharing the COVID virus or the sequence could have meant more and more people death and economical damage. I don't have numbers in mind, but I'm sure people have looked into that. Yes.
Thank you, Christine. Now we'll go with the next question. In the case of viruses and pathogens, DSI policy could vary depending on the situation. But in the case of endemic flora and fauna, could have the multilateral approach proposed in the current conversations? And this might be very related to our biodiversity question before. So perhaps this is one of the questions that we need to address in our next discussions. Okay.
So I can follow up with the next question. So to expand on the question about how to allocate benefits from a decoupled system, what do the panelists think the benefits should mainly be used for? Capacity development to use DSI, biodiversity science more generally, support to IPLCs, payment for ecosystem services, or should benefits be earmarked for use in the sector where they are generated, for example, health, food and agriculture, conservation, etc.? So the short question, what do the panelists think the benefits should be used for? Manuela.
Well, one of the needs I can talk about, it's capacity development to use DSI and resources for enabling our researchers from the developing countries to use the DSI in the same way and with the same capacity of the countries from the developed countries. And another issue that is very important is also to support the indigenous population and local communities, so they also can use DSI and take advantage of this technology, just for starting the discussion.
Would anyone else like to follow up?
I suppose I'll just add to that. I agree with what Manuela was just saying, but I think that is going to be one of the next big questions on, and Andrew was alluding to this, on once we thought about collecting the benefits, how do we distribute them? All I would add to that is I think there's an important next stage in this process around parties continuing to engage with stakeholders like scientists, and Manuela just pointed out some of the needs for capacity development around the use of DSI for researchers around the world, but also other stakeholders groups as well. And I think that is really going to be one of the big next steps working forward on this.
Thank you, David. We have one last question. We've been organizing all your questions because we want to answer them, and we will use it as input for keep on working on the discussion of this very important subject. The last question is for Derek. Options with payment for access will considerably decrease cross-platform use and research with DSI. I wonder how much and how often. Does it depend on the amount to be paid and how easy the transactions are?
Yes, thanks for that question. It's absolutely, it does depend on the amount that would be paid and how easy the transactions are. As I tried to explain at the beginning of my my presentation, it's very difficult to think through at this point what those there's a wide range of how of sort of very specific details and options as to how that can be configured. So we found it very difficult to make those sort of projections on how much would be paid and how easy the transactions are, particularly that latter bit. And we spoke to a number of, as a sort of an economic research team, we spoke to a number of scientists, including some of the people in today's webinar, about trying to get a sense of what these transactions could involve, their time, and then saying, okay, well, that also time, that time has a monetary value as well. So we arrive at this sort of finding without having counted all the numbers. We'll admit that, because we think that counting the numbers is too difficult to do, but there is enough indication from trying to think through the options and think through the numbers in terms of particularly how those transactions would work, what time it would take people in particular to arrive at our, maybe it's more of a hypothesis, sort of thinking forward prospectively. What does this mean? That the options that have payment for access would considerably decrease the use, and especially the cross-platform use and research with DSI. So we think that that's it does depend on those factors. So it's also not you can look at it with some illustrative numbers or ideas, and you sort of quickly arrive at that type of conclusion.
Thank you, Derek. So with that, I'm sorry to say that our time together has come to a close. I would like to remind everyone that today's webinar was recorded and it will be made available on the Global Plant Council's YouTube channel and on the DSI website. We encourage you all to share this recording and share with your network and keep this discussion going. So on behalf of Martha and all the organisers, I would like to give a big thank you to all our expert panel members for their contributions to this webinar today. I would also like to thank our translators, Cynthia Diaz and Helen Jimenez, who have been here translating everything that we say. Thank you very much. Also, thank you to Zara Lana, Isabel Mendoza, and Priscilla Mugo, and all their associates for their work and assistance in organising today's webinar. And finally, I would like to thank you, the audience, for your questions and attendance and interest in this topic. Take care, everybody.